Welcome to the PubMed Daily Review for March 16. Today we scanned 1074 new open access papers. Here are the top 4 selected for detailed analysis. Trend 1 of 5, Patient Outcomes and Medical Interventions. These papers investigate how medical actions and patient characteristics influence health results. This cluster covers 237 papers. Representative work includes the relationship between the American Society of Anesthesiologists' physical status classification and patient outcomes, a scoping review protocol, changes in passive muscle stiffness following biceps femoris strain injury in track and field athletes, cross-sectional and longitudinal analyses, and efficacy and safety of Vibrance add-on therapy for persistent overactive bladder symptoms in benign prostatic hyperplasia patients with alpha-1 blocker treatment, a multi-center prospective randomized controlled study, VATON study. Trend 2 of 5, Genetics, Evolution, and Immunity. These papers explore how genetic mechanisms, evolutionary adaptations, and immune system components interact to regulate DNA and RNA processes. This cluster covers 268 papers. Representative work includes risk assessment and management of Brucella canis introduction via commercial dog imports into France. Correction to SSX2 is a novel DNA-binding protein that antagonizes polycomb group body formation and gene repression, and diversity and evolution of archaeal immune strategies. Trend 3 of 5, Health, Biomarkers, and Interventions. This trend explores how biological markers and interventions impact health outcomes across diverse conditions. This cluster covers 248 papers. Representative work includes physiological responses to virtual reality-based stress regulation and relaxation interventions, a systematic review, clinical correlates of compliance, appeasement and resistance in command hallucinations, a systematic review, and exploring pain researcher and clinician perceptions of complementary, alternative, and integrative medicine, a large-scale, international cross-sectional survey. Trend 4 of 5, Ecosystems, Data, and Engineering. This trend unites research on complex systems, leveraging data analysis and engineering principles for understanding and manipulation. This cluster covers 189 papers. Representative work includes structural mass spectrometry techniques for characterization of plant and algal proteins, widespread higher soil respiration rates at nighttime than daytime across global forest ecosystems, and a surrogate calibrated updating method for logistic regression with missing covariates. Trend 5 of 5, Cancer Research and Therapeutics. This trend focuses on understanding cancer biology and developing innovative treatments and diagnostic approaches. This cluster covers 132 papers. Representative work includes editor's note on critical role for p53 serine 15 phosphorylation in stimulating transactivation at p53 responsive promoters, the features and prognostic value of ARID1A mutation and protein expression in endometrial cancer of no specific molecular profile, NSMP, subtype, a retrospective study in a large Chinese cohort, and whole abdominal radiotherapy in bone and soft tissue sarcomas, indications, techniques, clinical outcomes, and future directions. Up next, our first paper out of 4, Structure-Guided Design of a PfCyRPA-based Vaccine Against Blood-Stage Malaria by Nawzada Lam and Matthew K. Higgins from Biochemistry, Dorothy Crowfoot Hodgkin Building, University of Oxford, South Parks Road, Oxford, OX1, 3QU UK. Let's break down its key contributions. Malaria remains a devastating global health challenge, with millions of cases and hundreds of thousands of deaths each year. While progress has been made with pre-erythrocytic vaccines, we urgently need more effective blood-stage vaccines to prevent disease and transmission. The PfCyRPA complex is critical for the parasite's entry into red blood cells, and its components are prime targets for next-generation vaccines. To tackle this, researchers focused on PfCyRPA, a key protein involved in parasite invasion. Using structural information, they rationally designed a new immunogen, called PfCyRPA-EM, which specifically mimics the parts of PfCyRPA that trigger parasite growth-inhibiting antibodies. This engineered immunogen was then conjugated to nanoparticles and tested in rats to assess its ability to elicit a strong immune response. The results were promising. Rats immunized with the PfCyRPA nanoparticle construct developed about twofold higher levels of antibodies that bind to PfCyRPA. Crucially, these antibodies were also more effective, showing approximately twofold lower concentrations needed for 50% parasite growth inhibition. Furthermore, the new immunogen was more stable and easier to produce. This study demonstrates the power of structure-guided design in creating improved vaccine candidates. The novel PfCyRPA immunogen shows significant potential for developing more effective blood-stage malaria vaccines. Its enhanced stability and immunogenicity make it a strong candidate for clinical trials, potentially in combination with other vaccine components for even greater protection. Up next, our second paper out of 4. Gut microbiota modulation via repeated donor fecal transplantation improves motor and gastrointestinal symptoms in drug-naïve Parkinson's disease. A randomized phase 2 trial by Rui Zhang and Xuejing Wang from Neurology, the first affiliated hospital of Sun Yat-sen University, Guangzhou, Guangdong, China. Let's break down its key contributions. Parkinson's disease is a progressive condition affecting movement and daily life. While we have treatments, they mainly manage symptoms and don't stop the disease from progressing. Emerging research points to a strong connection between the gut and the brain, suggesting that the gut microbiome might play a key role in Parkinson's. This study investigates if transplanting fecal matter from healthy donors could be a new way to help patients, especially those who haven't started conventional medication yet. To test this idea, researchers conducted a rigorous study. They enrolled 72 patients with Parkinson's who were not yet on medication. These patients were randomly assigned to receive either fecal transplants from healthy donors or transplants using their own stool. The donor transplants were given in repeated cycles over several weeks. The main goal was to see if motor symptoms improved after 35 weeks, but they also looked closely at gut health, quality of life, and changes in the gut bacteria. The results were quite striking. Patients receiving donor fecal transplants showed a significant improvement in motor symptoms, with their UPDRS-3 scores dropping by an average of 3.8 points, compared to a negligible change in the control group. This difference was highly statistically significant. Furthermore, constipation, a common and debilitating symptom, was dramatically reduced in the donor FMT group. The study also noted positive changes in the gut microbiome and found the treatment to be safe and well tolerated, with only mild side effects. So, what does this mean for patients? This study provides strong evidence that fecal microbiota transplantation could be a promising new treatment for Parkinson's disease, particularly for those in the early stages. It suggests that by targeting the gut microbiome, we might be able to influence the course of the disease itself. While these findings are exciting, more research is needed to refine these treatments and fully understand how they work and their long-term impact. Up next, our third paper out of 4, integrating single-cell and spatial transcriptomics with pan-cancer bulk sequencing identifies OSR2 as a multifaceted biomarker for prognosis and tumor immunity by Zhihui Huang and Weran Wei from RUM, West China Hospital, Sichuan University, Chengdu, China. Let's break down its key contributions. The development and progression of cancer are complex, involving numerous genetic changes and distinct tumor subtypes. While we've made strides in understanding these processes, identifying reliable biomarkers for prognosis and treatment remains a challenge. OSR2, a gene known for its role in development, is increasingly implicated in cancer, but its full potential as a clinical tool is yet to be explored. To tackle this, researchers performed a broad analysis using large public datasets like TCGA, SIDE, and CROST. They employed advanced techniques including single-cell and spatial transcriptomics, alongside bulk sequencing. This allowed them to examine OSR2's expression patterns, its link to patient survival, and its intricate relationship with the tumor's immune landscape and other molecular features. The results were quite striking. OSR2 expression was found to be significantly altered, being upregulated in 13 cancer types and downregulated in 19 others. Importantly, higher OSR2 levels were linked to poorer patient prognosis in several cancers, such as gliomas and kidney cancer. Furthermore, OSR2 showed complex associations with tumor heterogeneity markers like TMB and with various components of the tumor immune microenvironment. So, what does this mean for the clinic? This study highlights OSR2 as a promising pan-cancer biomarker, offering prognostic insights and potentially guiding immunotherapy strategies. While these findings are exciting, further investigation into OSR2's precise molecular functions in cancer is crucial. Ultimately, this could pave the way for novel therapeutic approaches targeting OSR2. Up next, our fourth paper out of 4, gut microbiota dysbiosis and metabolite imbalance mediate diabetic kidney disease inflammation. Mechanisms and intervention strategies targeting gut-kidney axis and NF-kappa B/NLRP3 pathways by Hao Liang and Jian Ma from Heilongjiang University of Chinese Medicine, Harbin, Heilongjiang, 150040, People's Republic of China. Let's break down its key contributions. Diabetic kidney disease, or DKD, is a devastating complication affecting millions worldwide. Despite advances in diabetes management, DKD continues to progress, often leading to kidney failure. This suggests we're missing key pieces of the puzzle. Recent research highlights the gut-kidney axis, proposing that the trillions of microbes in our gut play a significant role in kidney health and disease. This paper is a comprehensive review, systematically analyzing existing research. It focuses on the intricate connection between gut bacteria, the substances they produce, and the development of inflammation in diabetic kidney disease. The review delves into how an imbalanced gut microbiome can compromise the intestinal barrier and activate key inflammatory pathways like NF-kappa B and NLRP3, ultimately harming the kidneys. The review found compelling evidence that in DKD, the gut microbiome is significantly altered. Beneficial bacteria that produce short-chain fatty acids, or SCFAs, are reduced, leading to lower SCFA levels in patients. Conversely, harmful metabolites like TMAO are elevated, with levels above 8 micromoles per liter significantly increasing the risk of end-stage renal disease. This imbalance impairs the gut barrier and fuels inflammation through specific molecular pathways. This research underscores the critical role of the gut-kidney axis in diabetic kidney disease, opening up exciting new avenues for treatment. Interventions targeting this axis, including traditional Chinese medicine, SGLT2 inhibitors, and probiotics, are showing promise in managing inflammation and improving kidney function. While these findings are encouraging, further studies are essential to refine these therapies and fully harness their potential for DKD patients.